Hello, I'm Sally Dunwoodie from the Chang Cardiac Research Institute. Congenital heart disease arises when there are structural abnormalities in the heart as it's forming. And essentially we're talking about babies being born with heart defects. The most common is referred to as a hole in the heart, and this describes holes in the wall or the septum of the heart which allows deoxygenated blood and oxygenated blood to mix. The problem with this is the blood that's sent around the body is not fully oxygenated, and this is not optimal for organ function. Heart defects are the most common form of birth defect in Australia and indeed worldwide. 3% of babies are born with some sort of defect, and a third of those are born with heart defects. In Australia, 1,500 babies are born each year with severe heart defects, and these babies require critical cardiac care. In addition, another 3,500 babies are born with milder heart defects. Now, these defects may not be picked up at birth, and they're identified later on in life when symptoms arise. Now, due to improvements in cardiac care, 85% of babies born with heart defects now survive, which of course is a wonderful thing, but does raise the issue that for them, when they have children, that their children are actually 10 times more likely to be born with heart defects. And we estimate in Australia there are 50,000 individuals over the age of 16 who actually have congenital heart defects. Congenital heart disease is caused by gene defects. In only a few cases have genes been identified and the defects been identified to explain how heart defects arise in babies. If we don't know what gene is defective and how it's caused the heart defect in an individual, we can't genetically define that congenital heart disease in that individual, and we can't advise the families of their future risk of having a child with a heart defect. Our group has made an important finding to help us identify genes that are associated with causing congenital heart disease. Now, it's always been assumed that it's the genes that are active in the heart while it's being formed are those most likely to cause congenital heart defects. And that's not a bad assumption. But what we found is that a gene called cyto2 is actually required for the normal formation of the heart, but that it is not active in the heart, it's active elsewhere in the embryo at an earlier time point during development. And if this cyto2 gene is defective outside of the heart tissue, it can actually impact on the formation of the heart and lead to heart defects. This research is important because it enables us to think about different sorts of genes that may be important in causing congenital heart disease. So at the moment on our list, the genes that we think are involved in causing congenital heart disease are those that are active in the heart while it forms. But we now need to add genes such as cyto2 to this list, genes that are not active in the heart, but indeed are active elsewhere in the embryo, but still, when defective, impact on normal heart development. This finding is important because it could lead to more cases of congenital heart disease being genetically identified. This is important for the families, for the patients, and for the families to understand what their risk is in the future of having another child born with a heart defect.